One, two, three, go. Hi guys, our topic for today is cell cycle and its malfunctions.
Hi guys, before we start, I have a fun fact for you. Did you know that the fastest dividing human cells, like those in your intestines, can complete a full cell cycle in as little as 10 hours? Compare that to a slow dividing cell like a liver cell, which might take over a year. It's like a race where some cells are sprinters and others are marathoners.
Let's now explore cell cycle. The cell cycle is the process cells go through grow, replicate DNA, and divide. When the cell cycle is disrupted, it can lead to diseases like cancer. It ensures proper growth, tissue, repair, and reproduction.
This is an example of cell cycle. Number 1. Interphase. The preparation of phase. G1, S, G2. 2. Mitotic phase. Prophase, metaphase, anaphase, and telophase.
What is mitosis? Mitosis is a cell's way of making copies of itself. It happens when a cell needs to grow, repair itself, or replace old or damaged cells. The cell prepares for mitosis by getting ready. It makes sure it has everything it needs to divide into two new cells. So in other words, mitosis is the process by which a cell prepares and divides to produce two identical cells, ensuring growth, repair, and replacement in the body.
Here are the 6 stages of mitosis. First, interphase. Second, prophase. Third, metaphase. Fourth, anaphase. Fifth, telophase. And lastly,
Interphase. Before mitosis begins, the cell goes through a period called interphase. During interphase, the cell grows, duplicates its DNA, and prepares for division. Although not technically part of mitosis, interphase is an essential precursor to the process.
Let's proceed to prophase. This is the first stage of mitosis. During prophase, the cell prepares for division by condensing chromatin into visible chromosomes, breaking down the nuclear envelope and forming spindle fibers to organize chromosome movement.
metaphase. During metaphase, the chromosomes line up along the metaphase plate, which is an imaginary plate equidistant from two poles of the cells. This alignment ensures that each daughter cell will receive the same number and types of chromosomes.
Anaphase. Anaphase is characterized by the separation of sister chromatins. The spindle fibers attached to the centromeres shorten, pulling the sister chromatins apart and moving them toward opposite poles of the cell.
Telophase. So during telophase, the final stage of mitosis, chromosomes reach opposite ends of the cell. New nuclear envelopes form around them. The chromosomes unwind into chromatin and the spindle fibers break down, preparing the cell for cytokinesis.
Now, let's talk about cytokinesis. Cytokinesis is the final stage of cell division, during which the cytoplasm of the cell is divided into two daughter cells. In animal cells, this process is accomplished by a cleavage farming, which finishes the cell membrane inward until the cell is divided into two.
After cytokinesis, two daughter cells enter interphase, and the cycle may begin again. Each daughter cell produced by mitosis is genetically identical to the parent cell and contains the same number of chromosomes. Mitosis is essential for the growth, development, tissue repair, and asexual reproduction in multicellular organisms.
So by studying the cell cycle, we can better understand how this malfunction occur and develop strategies to prevent or treat such conditions. The key to understanding the different disorders and diseases as a result of the malfunction of cells lies in our knowledge of the cell cycle. Failure to regulate cell activities may result to various diseases and disorders.
Because of the malfunction of the cell cycle, cancer occurs. Let us define what is cancer. Cancer refers to a group of diseases characterized by uncontrolled and abnormal cell division. It occurs when there is a disruption in the cell cycle. Instead of stopping and starting at appropriate points, cancerous cells divide continuously until a disorganized solid mass of cells called tumor is formed.
What causes cancer? Cancer is caused mainly by changes or mutation to the DNA within the cells.
So what is genetic disorders? It is a change in the number or structure of chromosomes can dramatically change the traits of an organism and can cause serious problems. Abnormal chromosomes most often happens as a result of an error during cell division.
Now, let us explore about chromosome abnormalities. Chromosome abnormalities often happen due to one or more of these. For example, errors during dividing of sex cells in meiosis. Second, errors during dividing of other cells in mitosis. The third one, exposure to the substances that can cause birth defects. That is called teratogens.
Karyotyping. Karyotyping is the process by which photographs of chromosomes are taken in order to determine the chromosome complement of an individual, including the numbers of chromosomes and any abnormalities.
So this is an example of normal human karyotype, male karyotype at the left and female karyotype at the right. A male karyotype typically has 46 chromosomes, including one X and one Y sex chromosome, or XY. While a female karyotype also has 46 chromosomes but includes two XX chromosome, or XX.
Due to the malfunction of cell cycle, numerical abnormality, also called aneuploidy, a condition which occurs when an individual has a missing chromosome from a pair, or monosomy, or has more than two chromosomes of a pair, trisomy or tetrasomy. For example, of chromosomal abnormalities under this category is include to the following.
Down syndrome. Down syndrome or Down's syndrome, also known as trisomy 21. It is a genetic disorder caused by the presence of all part of a third copy chromosome 21. It is usually associated with developmental delays, mild to moderate intellectual disability, and the characteristic physical features.
Turner syndrome occurs when part or all of an X chromosome is missing from most or all of the cells in a girl's body. A girl normally receives one X chromosome from each parent. The error that leads to the missing chromosome appears to happen during the formation of the egg or sperm.
Another disease that may occur is the Klinefelter syndrome, 47XXY. Klinefelter syndrome, also known as 47XXY, is a chromosome anomaly where a male has an extra X chromosome. These complications commonly include infertility and small, poorly functioning testicles.
Trisomy X syndrome. Trisomy X, also known as triple X syndrome and characterized by the karyotype 47XXX, is chromosome disorder in which a female has an extra copy of the X chromosome. It is relatively common and occurs in 1,000 females, but is rarely diagnosed. Fewer than 10% of those with the condition know they have it.
Trisomy 13. So let's proceed to Patau syndrome or trisomy 13. It is a clinical syndrome that occurs when all or some cells of the body contain an extra copy of chromosome 13. It is characterized by cleft lip, cleft palate, cerebral defects, anophthalmia, seminal creases, polydactyly, trigger thumbs, and capillary hemangiomata.
The next one is Edwards syndrome. Trisomy 18, also known as Edwards syndrome, is a genetic disorder caused by the presence of a third copy of all part of chromosome 18. Many parts of the body are affected. Babies are often born small and have heart defects. So in this part, babies are commonly affected.
Cre-du-Chat syndrome. Cre-du-Chat syndrome or 5p syndrome is a rare genetic disorder that occurs when a small section of chromosome 5 is missing. The condition is characterized by a high-pitched cry that sounds like a cat, delayed development, and distinctive facial features.